Okay, let's see. All right, then. So, good morning, everyone. So today we have the honor of having Dr. Eszter Boros giving us a lecture on the research that she has been doing over the years. So just to give you a little bit of background, Dr. Boros did her master's in science at the University of Zurich in Switzerland, where she designed and synthesized bifunctional complexes for nuclear targeting of cells with RNA emitters. Then she did her PhD at the University of British Columbia, where she focused on acyclic chelates for the radioactive imaging with radiometals. She moved on to a postdoctoral research fellowship at Harvard Medical School, I believe for Peter Caravan, who's actually going to come here to give a talk at the end of the semester. And so currently she is a professor, at the Stony Brook University. She will be relocating at the end of the semester to the University of Wisconsin-Madison. Her research interest lies in medicinal inorganic chemistry. Dr. Boros is a very prolific researcher, having published over 30 publications as an independent researcher, and she's highly funded with very prestigious grants, just to name a few: the NSF Career, an NIH-R01, an NIH-R35, and she has had many recognitions, most recently 2022, the Alfred Sloan Research Fellow in Chemistry, also 2022, Young Investigator in Medicinal Chemistry. So with that introduction, I turn it over to Dr. Boros.
Thank you so much, Arthur, and thank you so much for having me. This is really fun. If it was in person, it would be even more fun, I suppose, but I guess until then, Zoom will do. So I understand that you guys are learning about all sorts of cool stuff in metals and medicine and as part of this course. And so what I want to talk to you about today is work that's ongoing in my lab that's on a kind of really new project, like new chemistry to me, because I was mostly trained in radiochemistry and coordination chemistry and synthetic, organic, and inorganic chemistry during my PhD. And then for my postdoc, I did MRI contrast agent research, and the disease target was always cancer for us. But this is kind of a huge change in focus that I really only started when I started my faculty position at Stony Brook, and now will take with me to the University of Wisconsin-Madison. And so don't worry about the title, it's way too long. And I'm not sure if I'm going to be able to talk about titanium, because I really want to give you a good, as complete of a story as possible about our siderophore work and our antibiotics work. So just to give you, first of all, a quick glimpse of a possible opportunity, if you really like what you hear and see today, I highly recommend that you check out the CHOPS program. And so this is an opportunity at the University of Wisconsin-Madison to learn about the graduate program and how to apply successfully to our graduate program. You might know that the chemistry program at UW is definitely, depending on which path you're thinking of, but in inorganic, it's a top 10 program. It's very competitive, but also like really like world-class research is what we do there. So if you're interested in being part of that and you're not in grad school yet, but you're thinking of grad school, then I highly encourage you to apply. And so you can check that out here on the on this link here. Just laser pointer. So this is the link for the CHOPS program. And so this is like an all-inclusive trip that you can get to visit Madison, probably the best time of the year. It does get a little cold there, but as I said, yeah, it's a great opportunity. You can learn also about research opportunities in general. So with that, let me tell you a little bit about generally the research program in my lab. So we are straddling many different things and sort of bringing together many different corners of chemical research. And so we do some rare earth chemistry. And so we have a project in my lab where we take luminescent coordination complexes that are based on luminescent lanthanides, and we turn them into in vivo compatible optical imaging agents for cancer applications. We have another project in our lab that looks at the coordination chemistry of understudied early transition metal ions like scandium or titanium to turn them into useful radiopharmaceuticals for diagnostic imaging and radiotherapy, because we have access to radioactive isotopes that are either very good imaging isotopes, as you can see here on this picture here of a mouse that we have a tumor on and we're able to image the tumor on, or they have isotopes that are useful to create tissue damage in a very targeted fashion. And so, for instance, scandium also has a therapeutic nuclide, scandium-47, that allows us to impart damage to cancer tissues and then prolong survival in animals that are part of this cancer model. And then the third project in our lab kind of brings together aspects of bioinorganic chemistry and medicinal chemistry, where we're interested in tackling this big and emerging issue of resistance to antibiotics. And so infectious disease now, especially after the COVID pandemic, we know is an issue that we should no longer ignore and really think about how we can efficiently not just prevent, but treat it once it occurs. And so bacteria and resistant bacteria are really becoming a big problem. And so what kind of ties together everything in my lab is, because I was trained as a radiochemist, I kind of brought that with me into my own research program. Is every single one of our projects actually has a component that incorporates radioactive isotopes into our research. So in one of our projects, the isotope is actually producing photons that we use to excite these luminescent lanthanides here. In another project, we're using, of course, here the radioactive isotope as the main actor of imparting either a diagnostic modality or radiotherapy. Or then in our Siderophore project, in our antibiotics project, we were using radioactive versions of our antibiotics to better understand how they distribute in an in vivo system and how efficiently they're being uptaken by bacteria. And so what I want to tell you about today, which is, I think, going to be the main focus, is our project on what we call xenometallomycins. And so these are tools for imaging and also mainly the treatment of bacterial infections. And so this is already something I briefly touched on, but just to reiterate that really antibiotic resistance is a global public health threat in many countries. Like, for instance, I'm originally from Hungary. Clinicians like to overtreat with antibiotics, basically just throw the kitchen sink at patients that show up in the hospital, and that actually causes a lot of this emerging resistance. And the CDC estimates that the current death toll may only be under 1 million, only 700,000 worldwide annually, but by 2050, we could be looking at potentially even 10 million deaths caused by antibiotic resistance. And so antibiotic resistance is not just a human problem. The first time that I was confronted by this was actually when my dog, this is my late dog Bubbles, had recurring bacterial infections that he would acquire in his spine. And so as he got older, the regular course of antibiotics didn't work anymore. And so he had developed a resistant bacterial infection that then had to be treated with much more advanced measures and other types of combination antibiotics. And so this is really not just a human crisis, it's also, of course, a veterinary crisis. So how can we address this? And so to address this, as medicinal inorganic chemists or bioinorganic chemists, we think about metallic nutrients, right? So metal ions that are important nutrients to our body. And so how, for instance, that would be iron. And so when we take up iron, we have the advantage to eat our iron sources, right? Depending on what kind of iron sources we prefer, salad or steak or whatnot, we can take up iron in different forms, iron sulfur clusters or iron heme, and we can digest that in our stomachs, and then through the intestinal lumen from iron 3 to iron 2, can then take up the iron and either store it as ferritin or circulate it as iron transferrin, where the iron is in both cases actually as an iron 3+ preserved, and this is sort of the mobile version of iron, and this is the stored version of iron as ferritin. And so bacteria don't have the luxury to, go to the grocery store and buy themselves a salad or a steak, but what they typically do, especially if they are in an environment that is fairly hostile, like our bodies, is that they do piracy. And so they steal the iron from the host's resources. And so the host iron is usually in this transferrin. And so what bacteria do is this one pathway of iron sequestration and uptake is they produce these small molecules called siderophores. And these are really nice custom-built natural products that have very, very, very high affinity to iron-3. And so they excrete this into their immediate surroundings. And this siderophore will then very selectively bind to the very little iron or transchelate the iron that is even present in something like transferrin. And then using active transmembrane transport, uptake the iron through their membrane. And so this is an example that's a gram negative, where we also have a periplasm. So the uptake mechanism is a little bit more complex. So we have multiple transporters that very selectively recognize this complex, internalize it, and once it arrives in the cytoplasm, the iron then is either dissociated by a redox mechanism, so reducing it to iron 2, or the siderophore is modified chemically, which also results in a release of the iron. The iron then enters the electron transport machinery and synthesis. So electron transport proteins like iron sulfur clusters are synthesized, and the siderophore can either be degraded or recirculated. And so there's been this hypothesis around that these siderophores, because they're endogenous to the bacteria and we don't actually have them as hosts, could be means to deliver also antibiotics to the bacteria and then very selectively kill them in a very complex environment. And so this is a great idea. And typically when you have a great idea, somebody else had it before you, and that other person is actually nature. And so nature has been doing this for millions of years very, very efficiently. And so these two natural products are examples for what we call sideromycins. And so sideromycins are siderophore antibiotics that occur naturally. And so these natural products, you can see in the red, have an iron binding domain, as you can see here and here, salmicin and albomycin. And they have a short linker here and here that connects them to this blue portion of these molecules, these natural products that actually are the antibiotic portion. And so these molecules can impart ways to impose toxicity to the bacteria that internalize these molecules. And so these molecules are actually being synthesized by bacteria that compete for tightly controlled iron resources with other bacteria. And because those bacteria that synthesize these molecules don't actually have the means to degrade these molecules, it doesn't become toxic to them. However, all other bacteria they're competing with very efficiently and quickly degrade and cleave the green linkers here. And that will then release the blue portion of the molecule and become very quickly toxic and deadly for the competing species. And so this was a concept that was actually, and these molecules were discovered quite a while ago, almost a century ago. And their efficacy to actually kill bacteria was so good that in the Soviet Union in the 50s, albomycin was actually produced on a large scale to treat bacterial infections. And so it's actually active against pneumonia strains and staph strains. And so that makes it, of course, a molecule of interest. However, you can tell that if you've ever done a chiral synthesis before, which probably some of us have and others have the fortune to not have to do, this is a very difficult molecule to chemically make. And so if we only use biosynthesis to make this, we're very quickly going to run out of compound to actually treat a large number of people. And so this natural inspiration has inspired a lot of work in this field. And so here are some examples of people who have worked in this area before us. And so for instance, Marvin Miller at Notre Dame, who's now an emeritus professor, developed a variety of catechol-based systems and also hydroxamate-based systems that he linked to beta-lactams and was able to show that these can be quite efficacious to treat bacterial infections. And then Liz Nolan at MIT more recently has also shown that these functionalized enterobactin molecules are very good at very selectively treating pathogenic E. coli strains. And so what's really exciting for our field is actually that after all the decade-long work from many labs, a compound that mirrors this concept of bringing together a siderophore component and a synthetic antibiotic has actually made it into the clinical realm. And so Fatroja was approved in I believe, 2019 as the first siderophore based antibiotic that made it into humans and now can be used to treat complicated UTIs. And so there's a lot of excitement around this concept, but there's a lot of limitations too. And one of the limitations is that when these molecules are combined with iron, they can either lose their efficacy or they can then suddenly only turn into molecules that have very, very selective efficacy only in very few select strains. And so it turns out that if they're co-fed with the iron, that sort of offsets their potency. And so we were thinking, well, if this is a functional concept, how can we still convince the bacteria to take up these molecules without actually giving them also something that's useful? And so we found some other literature that used a metal ion that looks very much like iron and behaves like iron for the most part structurally, which is gallium. And so there's actually also quite a bit of literature on the antibiotic activity of gallium salts. And so reason for that is that the gallium 3+ and iron 3+ look very, very similar to nature. And so in my introduction, I talked about how iron 3 is the oxidation state that is used by nature to actually metabolize iron and also store iron. And so therefore, that's also the predominant oxidation state that uptake occurs in bacterial systems. And so what you see here is that a gallium version of ferrochrome, which is another bacterial siderophore, was used to co-crystallize transporter FHUD, that's a transmembrane transporter in E. coli that takes up this ferrochrome or ferrochrome-like metal complexes. And so you can see that this gallium complex works even for a crystallographic surrogate quite well. This also resulted in research that was done by a group from the University of Washington that was able to show that for ocular bacterial infections in rabbits, gallium desferrioxamine complexes could actually be quite efficacious. And they even managed to actually progress the work that they did with gallium citrate, not just from mice that you can see the data for right here. So this is a survival plot where you see percent of animals survived, and this is the time from 0 point of start of infection and treatment over 72 hours. They were able to show that a gallium citrate formulation led to prolonged survival in mice that were given Pseudomonas infection in their lungs. They even were able to progress from this animal study even to in vivo studies in humans who have cystic fibrosis and usually suffer from complicated Pseudomonas infections in their lungs. And so taking these two pieces of information and these two concepts together, we proposed when I started my group at Stony Brook to sort of use this combination approach that we were hoping would really amplify the potency of these compounds. And so we wanted to take the siderophore, combine it much like nature does with an antibiotic to impart therapy using the siderophore mediated transport. But we wanted to include a metal ion that could also exert therapeutic efficacy instead of being actually a nutrient that the bacteria need. And we also wanted to, of course, pick a metal that would have a radioactive isotope available to us that we could use to study the bacterial uptake with. And so that sort of combines what we call the classical medicinal inorganic chemistry concepts that build on molecular design, look at in vitro activity, elucidation of mechanism of action, and cross-references it with nuclear medicine strategies, where we look at uptake efficiency and pharmacokinetics using radioactive versions of these molecules. And so this was the concept, again, to reiterate. We wanted to have our gallium complex that then would internalize using the active transmembrane transport that the siderophores use. And because the gallium doesn't actually do redox chemistry like iron does, it would no longer be useful to the bacteria, but by the time it would already accumulate in the cytoplasm. And at the same time, we would also deliver antibiotics. And so at the same time, we could also make these molecules radioactive and study quantitatively the uptake of these systems. And so the isotopes we wanted to use were gallium-67, which is a fairly long half-life SPECT isotope. And then eventually we were also thinking of doing PET imaging with the shorter-lived gallium-68. And so when we embarked on this project, we selected a small selection of siderophores that we wanted to start with to start to test this hypothesis. And so we sided with the linear dysferrochrome and the desferrioxamine because that was the closest we could get to these two natural products that already are using this sideromycin concept. And the antibiotic we used was ciprofloxacin. Ciprofloxacin requires cytoplasmic delivery to be active. And so in order to guarantee that we were able to all the way deliver our gallium cargo as well as the antibiotic, we used the cytoplasmic active antibiotic. And so this was the inspiration for the chemistry. So these are the two natural products. This is the desferrioxamine based one, and this is the linear desferrochrome based one. And these were the two target molecules that we envisioned synthesizing. And so this is the, as you can see, the salmicin mimic, and this is the albomycin mimic. And so two students in my group, an undergraduate, Chloe, who in the meanwhile is on path to becoming a surgeon at Tufts University, and Apurva, who was the second PhD in my group and now is doing a postdoc at UCSF, took on this project and synthesized these respective molecules. And so the synthesis, of course, is challenging, but once we finish the synthesis, we need to figure out a way to test these systems. And so a most common way of testing these in vitro is in a minimum inhibitory concentration assay. And just to give you an idea for what this looks like, I have a schematic here for you. And so we typically use these 96 well plates where we have a decreasing concentration. And so here we have high concentration to low concentration of the agent we're looking to test for its ability to inhibit bacterial growth. And then we incubate every single one of these wells with the same amount of bacteria. And so as we decrease the concentration of the antibiotic, at some point we fall below the minimum inhibitory concentration of this antibiotic, and then bacterial growth will pick up and then increase as we further decrease the concentration of the antibiotic. And so schematically, this is what it looks like. The way we plot this data is like this. And so this is sort of a plot version of the well plate, right? So this is the optical density at 600, which measures the bacterial growth. And then on the x-axis here, we have the concentration of the antibiotic. And so ciprofloxacin, which is an FDA-approved antibiotic that I have been prescribed to as well. So possibly some of you have as well, including also my dog has gotten this prescribed, I suppose, too. So, you know, humans and dogs not so different in terms of the responsiveness to antibiotics. And so Cipro, for instance, against E. coli is quite effective. And so this is just our garden variety E. coli strain. And so the first thing that Chloe and Apurva tested were the gallium and iron desferrioxamine and gallium and iron LDFC linear dysferrochrome complexes. And so both the gallium desferrioxamine and the iron desferrioxamine complex actually didn't cause any growth inhibition. And neither did the linear dysferrochrome-based molecules. And so this was a little bit disappointing because we had hoped that at least the gallium complex would start to cause some growth inhibition. However, we hadn't actually incorporated the antibiotic moiety yet. And so we did that in the next step when Chloe's molecule and Apurva's molecule were ready to go to be tested. And so this is what the data looked like. And so this was our desferrioxamine-based molecule. And you can see the open symbols are the ligand by itself. Here, the filled symbol, that's sort of this brown is the iron complex, and the gallium complex in this light green and Cipro as a reference. And so unfortunately, it looks like by attaching this siderophore, we've actually made the antibiotic worse, at least by attaching the desferrioxamine. But something that we're starting to notice is that actually the iron complex across the board is worse than the ligand by itself and the gallium complex. Now, if we take a look at the linear dysferrochrome molecule, now we're seeing much improved performance. We're seeing that the open circles here, which is the ligand by itself, is just as potent as Cipro. And while the minimum inhibitory concentration breaking point is in the same place for the gallium complex, we see that the growth inhibition actually picks up a little bit earlier. We again see that the iron complex is much worse, right? Because we're doing this iron supplementation at the same time as we are actually also dosing our antibiotic. And so this was encouraging data. But we wanted to make sure that we're actually using the siderophore mediated transport mechanisms. And so this is sort of the hypothesis how these compounds work, at least in E. coli. In E. coli, the two different compounds have a different outer membrane transporter. And so while the ferrochrome-based molecule uses FHUA, the ferrioxamine-based molecule uses this FOXA transporter. Downstream, they use the same periplasmic and transmembrane transporter to the cytoplasm, but this first sort of initial uptake mechanism actually differs. And so here's again the data for the desferrioxamine-based compound. Here's the linear dysferrochrome-based compound. And so what we did was we purchased a specific mutant that actually has a non-functional FHUA transporter. And so by disrupting this FHUA transport mechanism, now what we should see is that in this type of strain, we only have one of the two compounds active. And we see that in the mutant, the desferrioxamine-based compound still works, but the ferrochrome-based compound is essentially useless. You can see that at very high concentrations, we see maybe some activity starting, but really it's nowhere close to what we saw before, right? And so this to us was enough confirmation that we were indeed efficiently targeting this active transmembrane transport, these active transmembrane transporters that are required to internalize these compounds selectively into bacteria. So where do we go from here? And so we made a compound that was pretty active, but we wanted it to be more active, of course, because right now we were just as good as Cipro. So how can we make this compound more active? And so this is essentially medicinal chemistry at its best. We look at a compound that has efficacy. And so what are some of the components that we don't have incorporated in our molecule that this molecule has? And so remember that again, we have the siderophore, so that looks the same. Then we have the antibiotic. And so we don't really want to use this antibiotic moiety because it's difficult to make. We want to revert to the FDA approved antibiotic, but we have a linker that differs very significantly. And so in this linker structure here, we have a serine, and the role of this serine is actually to be very selectively cleaved by proteases in the bacterial cytoplasm. And so taking inspiration from that molecule and from their activity against N-peptidases, Apoorva designed this molecule here. And so this molecule here, that is hard to make, but can be made, she named D5 because that's what she wanted to call it. And this molecule now incorporates, in addition to having a beta-alanine linker, also the serine linker that can be selectively cleaved by the N-peptidases. And so when she looked at the growth inhibition profile in E. coli, that was first a little bit disappointing. In E. coli, the compound D5 didn't do better. The gallium complex, again, was about as potent as the Cipro. You can see, again, the iron is a little bit worse. When we go to the mutant strain that doesn't have the functional FHUA transporter, we see that the potency decreases very significantly, which is exactly what we want to see. So we're still using these selective uptake mechanisms. In Pseudomonas, we've gotten much worse, so that's pretty disappointing. But we weren't going to give up because in staph, actually, we're we're able to increase the performance now. So we're now at a potency that was almost an order of magnitude better than the parent antibiotic ciprofloxacin. And so this was pretty exciting. And so where do we go from here? So the first thing that we wanted to make sure was that we actually needed the compound to be released cytoplasmically. And so if we would have a protease or N-peptidase degradation of this molecule, what we would release is what we call fragment A. That means that fragment A should only be exhibiting potency once it gets into the cytoplasm. And so when we tested fragment A against all of these bacterial strains, we saw that it didn't show any potency anywhere. And so we know that this is not a release that happens outside of the bacteria, but indeed actually happens inside of bacteria, because fragment A actually is not able to permeate any of these membranes. And so the next thing we did was radiochemical uptake experiments. And so the way this works in our lab is that when we conduct a radiochemical labeling experiment, because the radioactive isotope is fairly short-lived, we do that as the penultimate step. And so we build the whole molecule, and then we subject it to complexation with the radioactive isotope. Because the radioactive isotope is A, short-lived, and B, only present at very, very small concentrations, there aren't a whole lot of analytical techniques we can use for this. And so the primary analytical technique that we actually use is what we call radio HPLC. And so this is a conventional HPLC chromatogram, except for instead of a UV output, what you see here is actually counts. And so our HPLC instrument has a UV detector, and when the solvent comes out of the UV detector, it feeds directly into a radio detector. So that detector detects the amount of radioactivity that's coming through the tubing that's eluting off of our HPLC column. And this is what the chromatogram looks like. And then once we characterize these molecules, there's a whole lot we can do with it. We can look at complex stability in challenging environments, in broths, for instance, that we use to grow the bacteria in, or more interestingly, even we can look at the time-dependent uptake in bacteria. And so just to give you an idea for what this looks like, so this is the radio HPLC system that we have. This is where we do the chemistry. So it's quite bare bones. Anybody can do radiochemistry. It's not actually, especially with aqueous coordination chemistry and radioactive isotopes that are non-volatile, there's actually not that much you need. That's specialized equipment, except for a lot of lead to shield yourself from the radioactivity. And then here's the HPLC system that has a UV detector here and then a rad detector. And then we also have a TLC scanner that is nothing else but a detector that moves across the whole span of a TLC plate to measure the radioactivity in different spots on the TLC. So the first thing that Apurva did was just a simple radio labeling experiment. And so this is what atypical characterization looks like. And so we overlay the ligand HPLC trace in black with the radioactive HPLC trace in red. And so you can see that we have a small shift in the retention time that's diagnostic for the complexation, but we have a very rapid and efficient incorporation of the metal ion into the chelator, which is great. And so that's pretty straightforward. So now with the molecule in hand, we look at the efficiency of compound uptake over time. And so we do that by conducting bacterial incubation. And then we isolate the bacterial pellet and the supernatant, and we look at the counts of radioactivity in these different portions of our isolates. And so this is what we see. This is a typical uptake experiment. And so these are 3 different strains: E. coli, Pseudomonas, and Staph. And what you see in black is just gallium citrate. So that's our reference. That's not with a siderophore. And then gallium-67 D5 is our siderophore antibiotic conjugate labeled with gallium. And so what you can see in E. coli is that over time, as we continue to incubate, we see an increase in the uptake of this molecule. The same for Pseudomonas. And what we see, interestingly, is that, of course, the trend is preserved in Staph, but there we saw that the ciprofloxacin also was increasing over time. The citrate compound was also increasing in uptake over time. And so Apurva actually repeated this assay numerous times because this didn't seem right considering the other results. However, it turned out when we looked into literature is that actually gallium citrate is considered a siderophore metal complex for staph because staphyloferrin actually utilizes citrate as a synthetic component for the endogenous siderophore staphyloferrin. And so we could actually rationalize that this data indeed was real, that there was indeed uptake of gallium citrate that was selective for staph. And so the bacterial uptake data then motivated us to actually do the next experiment, which was an in vivo experiment. And so one of the things that's probably one of the most unique things about my lab, which in addition to, I suppose, a lot of the radioactive work, we also do the in vitro work and the in vivo work ourselves. And so what we did for this study is that actually Grace, who was a postdoc in the lab, and Apurva learned how to induce bacterial infections into the triceps muscle of mice. And so they took mice and they gave them a bacterial infection in one triceps, and in the other triceps, they induced an inflammation that had heat-killed bacteria. And then they injected the mice with the radioactively labeled compound and looked at the distribution after they sacrificed the mice and dissected and counted the organs on a radioactive gamma. And so this dissection result is how we usually represent things. And so on the y-axis, you have the percent injected dose per gram, which tells you how much of the injected dose per gram tissue is present. And then on the x-axis, you have all the tissues. And so this looks a little crowded. And so I'm going to draw your attention to the most important part of this plot, which is actually the control muscle and the infected muscle. And so what you see here is that the gallium citrate does not differentiate between the control and the infected muscle. However, the gallium D5 complex appears to selectively accumulate in the infected muscle, which is quite exciting. And so this motivated us to now move on and further investigate this compound. And so we were trying to figure out some ways how we could possibly and non-invasively actually monitor the bacterial infection in these mice as we were treating with our new antibiotic. And so what we sided with was to again use imaging, but not radioactive imaging in this case, but bioluminescence. And so we purchased a special strain of staph. And this special strain of staph that's called Zen29 exhibits bioluminescence. And so if you plate it out on an agar plate, this is what it looks like under the luminescence imaging equipment that we have at the animal facility. So it's very, very luminescent. So the plate glows. And so we decided that this was going to be a good way of monitoring active bacterial infection. And so, well, of course, because this was a different staph strain than what we had used so far, Apurva had to test again the potency of the antibiotics. First, the commercial one. And so she could do that actually by checking the luminescence, using the luminescence output of the bacteria as means of quantitating growth. And so in addition to looking at just the OD600, which is what we do with non-luminescent bacteria, we can actually just do a luminescence image and measurement here. And so here you can see just non-bacteria containing wells here. This is the little minus here, and all of these contain bacteria and are all equally luminescent. And then here's the growth inhibition experiment in triplicate. And so this is essentially a visual for the MIC experiment that I was explaining to you a little bit earlier. Here's the high concentration of Cipro. Going to the low concentration, you can see that nothing grows in the high concentration wells. And as we go to lower and lower concentrations of the Cipro, we're starting to see growth. And so you can cross-reference the region of interest analysis, the luminescence data with the OD600 data, and overlay it. And then you can see that the information that you get from both the UV vis and also the luminescence image match. Actually, it's also a really good diagnostic way of figuring out if you have contamination of your bacterial samples, because when we started with these experiments, we got the staph strain from a different lab in our building. And we found that the luminescence results didn't match the OD600 results on the same plate. And it turns out that it was actually contaminated. And so we had to buy it fresh from the commercial vendor. And so when Apurva tested gallium D5, this was a pretty exciting image because it showed, as you can see here, the bacteria with Cypros start to grow at a much higher concentration, whereas with D5, they start to grow at a much lower concentration of the antibiotic. And again, we can overlay the data, the OD600 data with the ROI data and see that it matches. And so with this information, this in vitro information in hand, we set out and we did the animal experiments. And so this is how we do the animal experiment. We inoculate the mice, again, in their triceps muscle with the Zen29 strain. And then we dose the mice twice with a known amount of our antibiotic intravenously. In between, we conduct bioluminescence imaging, and then we dose first right after we administered the infection, and then a second time 24 hours later. So this is an example of a mouse that didn't get any treatment that would be in any way helping with the infection. And so this really, really bright spot here is the bacterial infection in the mouse. And so we're using these BALB6 mice that are, no, not BALBs, BALB-C mice that are these white furry mice, and we have to actually shave their arms so that we can induce the infection, but also continue to image without any image perturbance. And so the saline animals actually expire quite quickly because they get an infection so bad that we have to actually put them down fairly quickly because they're in a lot of distress. If we treat with ciprofloxacin, which is again the FDA-approved compound, at a high dose, we have very good efficacy. And so you can see here the time of survival over the course of 7 days is 100%. So all of the mice in the cohort that gets the high dose of Cipro survive. However, if we're treating with a much lower dose of ciprofloxacin, we're not really seeing any improvement of the infection. And so those mice actually, unfortunately, also expire within a day. And so other things we can do in addition to monitoring the infection in vivo is we can sample the muscle and see how much of the bacteria remain in the muscle, sort of as a more quantitative measure. And we can measure that by after tissue homogenization, we can count the colony forming units. And so we do that as soon as we put the mice down. And so here is an example for what the plates look like. And so it's not even possible with that higher concentration plates to see the colony forming units because there's so many of them in these samples. So when we dosed with gallium D5, which is Apurva's compound, what we saw was an infection that started to actually clear up. And so we no longer had this really, really bright luminescence signal, but actually over time, over the course of multiple days, the infection actually completely cleared up in some of the mice. And you can see that at the equivalent dose as we used for the low dose Cipro mice, we actually had 100% survival. When we dosed at a considerably smaller dose, we still saw some survival of the animals over the course of 7 days, but certainly prolonged survival when compared to just the saline treatment. And so you can see also that the CFU measurements show that there's no active colonies here, whereas here with the low dose treatment, you can see that it wasn't quite a complete treatment. So there's a little bit of bacteria still remaining. However, this was still a very successful application of this concept. And so we call the compound now galbafloxacin, just inspired from Cipro. We think that this is like a nice demonstration of how we can apply sort of a medicinal inorganic chemistry concept and translate it into a compound that has in vivo efficacy. And so I showed you that we use molecular design that was inspired by nature to incorporate both the therapeutic metal, the diagnostic metal as well for studying pharmacokinetics and uptake, to then generate an antibiotic that is more efficacious than the FDA-approved compound that you can see here with respect to performance with an MIC value of 0.9 micromolar, and the mice at the same dose don't survive very well. However, we can actually give curative treatment at the same molar concentration of our new molecule, and we're able to show that the radioactive compound accumulated selectively in the infected muscle. And so this is basically the first complete part of the story, and I don't think I have time to talk about the second story, so I'm actually going to stop here. The person who did the majority of this work was Apurva Pandey, who was one of the founding members of my lab. She's doing a postdoc now at UCSF, looking at other roles of modifying iron in mammalian cells, ferroptosis, and Grace Ahn, who was the first postdoc in the lab. And she is now a staff member at Dana-Farber, doing radiochemistry and contract synthesis of radiopharmaceuticals. I didn't get a chance to talk about Angus's work on siderophores and titanium. Maybe some other time. People who are working on this project right now and are making great progress are Minhua and Owen, and also Axia. And you can see here, the lab is excited to be moving on to greener pastures, I suppose, in Wisconsin, and we're all pointing in the wrong direction, clearly. But I'm gonna, I guess, close here, and I'm happy to answer any questions you may have.
Well, a round of applause for Eszter. That was a fantastic talk. Thank you so much. So students, any questions? Feel free to get on your mic or write a question in the chat.
Yo tengo una, usted la podría traducir, es que no sé cómo preguntarla bien en inglés.
Sure. So Eszter, student has a question. He's going to say it in Spanish, but I will translate it.
Sure.
Go ahead.
Okay. Este tipo de partículas, si se quisiera utilizar para algún tipo de célula cancerosa, ¿sería básicamente la misma composición o se tendría que utilizar algún marcador dentro de la nanopartícula para que ésta se adhiera directamente dentro de la célula cancerígena, como un indicador de camino?
So Eszter, what Oscar wants to know is how can we translate this to an anti-cancer application? I know that you to some extent have been doing some of this work already for cancer, as far as using the radioactivity to do a theranostic type of study.
Yeah, so I mean, cancer applications with radioactive isotopes would be using different targeting vectors, right? And so here we're targeting an uptake mechanism that is unique to bacteria. And so when you want to target cancer, you have to look for a cell surface receptor that's overexpressed in cancer cells. And so we know of some of these, like some of these are pretty well known, like the prostate-specific membrane antigen or somatostatin, for some cancers. I mean, but it's still a very much like a developing field because we still need to better understand cancer biology to find targeting vectors for many different cancers that we can then attach a radioactive isotope to, that so that we can deliver that isotope very selectively to those cancer cells.
Oscar, does that? Yeah. Okay. Thanks, Oscar. Another question from a student. So Eszter, I do have a question myself. So I was just curious, and I know you mentioned it, that you said gallium in general has its own associated antibiotic properties, but is gallium in general known to be non-toxic to humans, such that this would be a really nice approach so that you have more selectivity and also the lack of the toxicity? Yeah,
so I mentioned this study by the group at the University of Washington. Let me go back to it. Let me just stop share real quick. So the gallium story is a really interesting one, because these guys at the University of Washington in the medical school did all these really interesting in vitro and in vivo studies. And they were able to show, so what they do in the human trial is they formulate the gallium nitrate with citrate to just try to keep it in solution, right? Otherwise the gallium would precipitate out actually because it's so hydrolytic that under physiological conditions, it would just form gallium hydroxide. And so they formulated with citrate and they infused the gallium citrate over days into people at a fairly high concentration. And so the good news of that study is that there aren't any adverse effects as far as they could see in these CF patients. The bad news of the study is that the study was actually completed and did not continue to phase 3 because they didn't see a statistically significant change in the patient outcomes for infection. And so I think actually the reason for that is that the gallium citrate is not sufficiently being uptaken by the bacteria. And so because what you need is for it to localize in the lungs first and then actually being internalized too. And so I think that's a big challenge, especially just if you formulate it with citrate. And so that's, I think, where the strength of these type of systems comes in, where you can be much more selective and target the pathogen with the siderophore that it would uptake already anyway, and then try to accumulate the iron in those bacteria more selectively and also deliver it more selectively, right? Because I feel like this gallium citrate treatment is a very brute force method of giving gallium, right? Like just imagine like you're sitting there for 2 days and you're just getting a gallium citrate infusion. I'm actually surprised that it doesn't have significant amounts of toxicity to the patients. The other thing to consider is that there is also the potential of using other metals than gallium. And so scandium and indium are actually sufficiently iron-like that they also impart some amount of antibiotic activity. And so that's something we've actually tested already, is the combination of gallium and indium, of indium and scandium complexes together with these antibiotics, because they still maintain some amount of affinity to these siderophores, but then have, of course, slightly different chemistry. And so we actually found that in many cases, the indium compounds can be just as toxic as the gallium compounds, sometimes in some cases actually a little bit more potent, which is really interesting. And this is super long-winded, but I'll just make an additional comment that while I think that this is like a promising avenue to go about trying to sort of double dose antibiotics or kind of double up antibiotics, we still really need to better understand why these compounds work and how these compounds work. And so that's now sort of the future work in my lab, besides trying to further optimize and expand the compounds that we're testing, is actually we're working on proteomics and metabolomics based probes that help us understand why the gallium works and how it works and what sort of its fate is once it gets into the cytoplasm. Because there are multiple hypotheses, but none of them has really been thoroughly studied and addressed, right? And so in order to make better compounds, we actually also just need to understand how and why these work in the first place.
Well, thank you so much, Esther. And to comment on a note that you made earlier, you're always welcome to come to Puerto Rico. So once you're officially settled in and you have time, it would be great to have you here in person.
I'd be delighted to come.
That's amazing. So a round of applause. Thank you so much for this brilliant talk. And I really appreciate it. All right, everyone, have a great day.
Thank you. Thanks for having me. Please reach out if you want to know more about the CHOPS program or anything about UW. Happy to always chat further offline too.
Great. All right. Bye-bye, everyone.